This is a video tutorial on DNA, base pairing, and transcription. Base pairing is when chemical bases are joined together by hydrogen bonds that connect complementary strands of DNA and RNA. The bases are adenine, thymine, cytosine, and guanine. When it comes to base pairing, both chemical bases are joined by phosphate sugars, and that means that only two of each of the four are complementary towards each other. The two that are complementary towards each other are adenine with thymine and cytosine with guanine. Part 2 is transcription. Messenger RNA, or mRNA, carries copies instructions from the genes for putting together amino acids into proteins. They carry messages from the DNA to the rest of the cell. Ribosomal RNA, or rRNA, make up major parts of the ribosomes. Proteins are put together there. Transfer RNA, or tRNA, transfers each amino acid to the ribosome as it is specified by coded messages in RNA. This is an amino acid chart, and it goes from first position second to third to find out the amino acid. The beginning and end points for the amino acid coordination are the promoter, which starts transcription, and the terminator, which ends transcription. For transcription, the whole strip of DNA are divided into threes. So for an example, there's TAC, TGA, and TCG. And for mRNA, it translates to AUG, ACU, and AGC. Then for tRNA, it goes over again to UAC, UGA, and UCG. And once it hits mRNA, the T is no longer there. The T only goes for DNA, but after that, the T is replaced by a U. Then for the amino acid that you find using the chart, you would find that it's TYR for the UAC, then it's STOP for UGA, and then it's SER for UCG, and that would be your amino acid line. Mutations are errors that occur during DNA replication. It is a change in the nucleotide sequence. When a mutation occurs, when you do the sequence and try to find out the sequence for the DNA, for an example, if there's the TAC, CGC, and a TCC, then the TAC goes to AUG for mRNA, then CGC goes into GCG, and for the TCC, the T goes into an A, the C as a mutation turns into a U, but the other C remains the same as in G. So when it comes to tRNA, it's going to be UAC for AUG, CGC for GCG, and UAC for AUG, which is the mutation when it really should be ACC. Yeah, I think that's right, ACC. Then for the amino acid, it goes to TYR for UAC, ARG for CGC, and STOP for